Good evening, everyone. I am Chrissy Kelly from Absent, and welcome to our 15th Tuesday evening webinar. I am delighted to have with me Nancy Rawson, a Vice President at the Menell Center. Nancy is a cell biologist, and I had the great pleasure of meeting Nancy a couple of years ago in Dresden. I've asked her to come this evening because she is so good at explaining things to people. So Nancy, welcome, and it's very nice to see you.
Great to be here.
We also have Federica Genovese, who is a postdoc at Menell, and her interest is the integration of olfaction and chemesthesis. So she is going to be here with her slides, and together we're going to be learning a little bit more about the coronavirus, how it gets into the system, what is the affinity of the coronavirus with the olfactory system. So just a couple of housekeeping notes. I'm sure you're going to have questions, as you always do every week. So please put them into the Q&A panel. Let's not use the chat bar if we possibly can. Use the Q&A bar, and I will be looking into that regularly, and I might even have to interrupt Fede or Nancy as they're describing something so that I can bring your questions in. So do feel free to ask questions. And I think that's pretty much it. So I think we're going to start now. Why don't we begin? Nancy, why don't you give us a brief overview, and then we'll turn it over to Fede and her slides.
Okay, great. Well, it's great to be here. Welcome. It's a pleasure to be able to share some of the great science going on around what's happening in the nose as a result of COVID. And so what we're going to do, I spent a lot of years studying the cell biology of smell and taste as well. And now I'm more administrative, but I love communicating the science that is going on at Menell and around the world. Really, it's been such a phenomenal global effort to advance this science. So I appreciate the opportunity to pull some of that together and help you kind of know what's going on inside your noses. So what we're going to do is Fede's going to share the slides. She'll be driving a few slides to get everybody on the same page and really help picture what's going on up there. And then I will, very good. And I'll give some of the presentation and then I'll turn it over to her. Does that sound good, Chris?
That sounds perfect. Thank you.
Okay. So we're going to dive into the nose and try to understand what this little guy, this little nasty COVID guy is doing up there. So why don't you go ahead to the next slide? We all are aware of the tremendous reports and disability and difficulties people have had with this virus that reduces our sense of smell. It can cause loss of smell. It can cause parosmia. It turns out it can even cause loss of taste and loss of chemical irritation, our ability to detect hot pepper and things. So what we're going to focus on initially here mostly is our sense of smell, because that's frankly what we know more about. And we're going to start in the nose because the virus is often gets into the nose through inhalation of droplets in the air. And if we are going to do this, we're going to shrink ourselves down and go on a journey. So think about getting really small and going up the nose, and what would you see? So this is what you would see. There's these turbinates, and at the top of your nose, right about here at the bridge of your nose, there's the cells that detect odors. And these cells are like, they're like finger processes. And you can see that in the blow up here. Again, the tissue inside your nose that occupies about a postage stamp area. And that houses about 6 million of these cells that detect odors. And the odor detecting cells are the ones that she's showing you now, olfactory neurons, we call them. And at the top of those cells, there are these hair-like projections called cilia that stick out into the mucus in your nose. And that's where the odor detection process happens. That's where the chemicals that come up your nose and anything else that comes up your nose, frankly, interacts with those we call cilia, and they're like little finger projections. And the other cell type, major cell type in this tissue, this olfactory epithelium, we call it, are these supporting cells. And the supporting cells are like a scaffold. So they're there and they're structurally creating a nice organized scaffold for the olfactory neurons to be aligned within and to support them. And we don't know a lot about these cells, frankly, because most of the time people spend studying the olfactory neurons. But we've suddenly become much more interested in these cells, and you'll find out why. But we do know that besides structural support, they play an important role in detoxification. So if you breathe in some toxic chemicals, there's enzymes in these cells that can break down those chemicals. And they're actually second only to the liver in their ability to detoxify chemicals. So it's pretty amazing. So they're very important cells in defense of that tissue and helping to protect it. So when odors interact, they send their messages to the brain. And you might think of the brain as a computer. So if the brain is a computer, the keyboard would be the olfactory epithelium, would be the cells that are sending the messages. And normally, you can imagine that the keyboard is in a very specific arrangement. You know where the A is, you know where the L is. And that's very important in communicating a message to the brain correctly. So when we think about what might cause smell loss, one of the first causes often is what we call obstructive. So if you have a cold and you can't breathe, the odors just can't get up to your nose to where those important cells are. So we call that obstructive loss. But we've learned very quickly that in COVID, those patients do not have nasal obstruction, yet they can still have a loss of smell. So we don't think that that's the cause for smell loss in COVID. So the second potential avenue for understanding this is what's going on in the olfactory epithelium. We call this sensory neural. And this is this structure of the tissue where the odors come in and they're detected by the cells and the message is sent to the brain. So we know that the message isn't getting sent to the brain, but we don't really know where the problem is, or we didn't know, right? We didn't know where to start. We knew that was something. And the first thing you might think of was the olfactory neurons. So I'll pass it over to Fede to talk about, in order to cause a damage, the viruses have to get into the cells. And in order to do that, I mean, they do that so that they can take all of this machinery that you find inside cells and hijack it. And they hijack it so that they can steal that machinery and use it to make more of themselves. So the viruses are really very clever. They have to get into the cell. And once they get into the cell, they use it to make more of themselves and kill the cell. So,
Nancy,
yes.
Can I just interrupt you and ask you, is that mechanism, that the way a virus gets into the cell to self-replicate, is that something that's universal to all viruses, or is that something that are we seeing something that's more peculiar to the coronavirus?
It's not peculiar to the coronavirus.
That's just viruses.
Right, that's a viral, that's how viruses attack cells.
Okay.
And the cells have this lipid bilayer membrane, which is very protective. It's very good. It's got like several layers, and it really is designed well to keep stuff out. So it's not that easy for things to get in. If it was, we would have a lot more problems, frankly. So Fede's going to take you through how it is that these viruses actually have managed to trick the system.
Yeah, so as Nancy said, the virus hijacked the cell machinery for making proteins. So what I would like you to do is to think at the coronavirus basically like a computer hacker. So able to get into every cell computer and write down its own code to replicate himself. But those viruses cannot do it with every single cell. They need a very specific one. And in the case of the coronavirus, they need the cell to express ACE2. So they need to have a specific code, or what we can think about is like a very poor password that the coronavirus can actually crack and being able to get in. Physically, what we think about, we can also think about ACE2 as an anchor. So the virus can anchor to ACE2 through its own spikes, and then from there can get inside the cells. So now the next question is, of course, now we see that there are olfactory loss. So, but where is ACE2 in the olfactory epithelium? Is it in the olfactory neurons? Is it in the supporting cells or somewhere else? So what it was done, this is actually quite recently, but it was also already shown in the past months, is that actually, if you look at this image and we look back at our previous scheme, we have in yellow our supporting cell, the scaffold, while here all this purple nuclei, those are the olfactory neurons. And so you see here on top in this light blue ACE2 protein. So ACE2, if we compare it back and we put our colors on our previous scheme, you can see that ACE2 is actually not on the cilia that Nancy talked about before, but on the villi, so the small finger of the supporting cells. So this is quite interesting because then it's basically making us think again to our computer, our olfactory system. So how breaking down the supporting cells will actually break down the communication between olfactory epithelium and the brain. Well, think about the infected olfactory epithelium, basically like just a bunch of keys from a keyboard. So without the scaffold holding all the keys in place together, you're actually unable to write down or send any information back to the brain. And this was also shown even more in an experiment in mouse model. So now get with me through those, which are a little bit complicated, but we can make it. So on top, you have the healthy epithelium. You have here, the first ones is basically an overview of it. And then here we have the yellow ones that we have seen before, which are the supporting cells. And here we have in green, the olfactory neurons. So basically imagine that we take a little piece of our olfactory epithelium, we cut it very, very thin, and then we stain the different kind of cell types that we want to see. So if you see here the healthy epithelium, already in this image, the structure is very well defined. So you have here on top the supporting cells, you can really see them nicely. And then here all the nuclei. So if we look at the colors that can help us even better, you can see here the supporting cells all there perfectly intact. While here with the olfactory neurons, you actually see them again lining down perfectly like in our keyboard. But also you can see here in bright green, the cilia, which are still all intact. So basically everything is ready to work. But what happens 4 days after the infection of the epithelium with the coronavirus? So here you can already see like a big, big change in the structure. So the nuclei are more distributed in an even way. You cannot really see this nice layer on top and so on. But then with the colors, then the differences really get striking. So you see our supporting cells in the healthy epithelium, they're basically not there anymore. And for our olfactory neurons, so you don't see any structure, but also the cilia that here you see perfectly well conserved are completely destroyed. And so here, those are basically just pieces of it hanging around. So those are kind of some example. Again, this is mouse model, but that can give us some ideas about how basically the lack of supporting cells, so the lack of the scaffold for our olfactory neurons, can actually completely destroy our olfactory epithelium and make it almost impossible for the olfactory system basically to detect the odors from the very beginning. So it's not a problem at this moment, basically in the brain. Yeah.
Rita, can I ask you, for anyone who's listening who's not familiar with the use of the mouse model to understand human olfaction, could you help people understand why it's such a good model? For someone that is not an uninitiated, they might say, well, okay, a mouse, but what does that tell me about my own personal sense of smell? And I know that there are good reasons why we use mice. So maybe you could tell us a little bit about that.
Yeah, so basically, we know already that the structure of our olfactory system, or at least of the olfactory epithelium in the mouse and in humans, is more or less the same. It's basically the same. And the very, very similar is also the expression of all the different proteins which are able to detect different odors and so on. Yes, we have a little bit more of olfactory receptors, so those proteins able to detect different parts of an odors in a mouse, but overall the system works exactly in the same way. So what the biggest difference I think we can say between humans and mouse is that the olfactory epithelium in both of them can actually regenerate. So it's one of the few neuronal population, so one of the few neurons that can actually regenerate through all our entire life. Mm-hmm. But in mouse, this mechanism is way quicker than in humans. And mostly it's also because the connection or the way that I have to do from the nose to connect back to the brain is also a little bit shorter than our way. So the space between here and here. But other than that, actually the mouse model is pretty good. Now, we can make it a little bit more complicated with the coronavirus and ACE2, because it seems to be that ACE2 in mouse is a little bit less sensitive to the coronavirus. So that's why in this model, actually, so the mouse was had a sort of transplant of human ACE2. So in this way, they could actually see how the coronavirus and the human ACE2 can bind together.
So this mouse had the human version of the ACE2 protein, so that it would be even better of a model for the human nose.
Yeah, and this kind of experiment actually have been repeated also on another species, which is a hamster, Syrian hamster, that I never heard before until now. But seems to be actually even better fitting with the dynamics of binding between ACE2 and the coronavirus. And actually in hamster, they see exactly the same thing. Another thing that it was interesting in the study with hamster is actually they saw that after being infected with the coronavirus, the epithelium was able to regenerate in 2 weeks, which is, of course, it's an hamster. So it's not really fitting with the human time for recovery, but again, it's actually showing that it's a good model because we saw recovery also in humans. And so the thing that also in the hamster we could see, and actually also in mouse, we could see some recovery. That's already tell us that the model is good.
Yeah.
And this is actually my last slide. And so with that, we can have more questions.
Thank you, Fede. Thank you very much.
Shall I close here?
Yep, you can
okay.
Close there.
Here we are.
Good. Right, well, I'm sure there are going to be questions, but maybe either of you could talk to us a little bit about, what does it mean that we know that the sense of smell does regenerate. Maybe you could kind of paint us a picture of what does that regeneration look like? What is the time scale? Obviously, we don't have the same metabolism as a mouse or a Syrian hamster, so for us, it's going to be slower. So let's talk about that a little bit because people always want to know more about that.
Yeah, in human, certainly the process is slower and less robust than it is in mouse. What we see, we don't know direct comparisons with respect to COVID, but for other types of damage to the epithelium in mouse, if you destroy the tissue with chemicals, for instance, they can recover pretty completely within a month. In a human, it might take years.
Yeah.
And I think depends a lot also on the kind of, how big is the damage and which kind of damage we have. And I guess that's also why with the, with COVID, the spectrum is so much wider. We have people that lose it, and then, I mean, I have family members that lost their sense of smell due to COVID, and in 2 weeks, they actually started re-smelling something again. But I heard about other family members of other people that needed way longer to doing it. So it really depends on how aggressively the virus was able to attack our olfactory epithelium.
Mm. Certainly in the patient group, we see, I think, a number of different kind of groupings of patients. We have people that recover really quickly. So they lose their sense of smell, and then within, a couple of weeks, they come back to us and they say they're absolutely fine, and then they leave the group. Then there are people who take a little bit longer to start recovering. They may have a period of sort of vague smells, but more of sort of hyposmia. They start to recover, and then they feel that they're well on the way to recovery. And then 4 or 5 months later, they develop parosmia. So that's a mystery. And in addition to that, thrown into the mix, and of course, very difficult in a group like the one we have, which is just about to hit 14,000. It's very difficult to kind of parse out, what's happening to which group. But what I can say is that there are also repeated reports of these fluctuations. And the fluctuations can be happening over one day. People's sense of smell might fluctuate. And when I say fluctuate, they report going back to nothing. And that might last for days or even weeks. And then the smell will come back again. And then, then they're going to get another fluctuation. So these fluctuations can be mixed of all different lengths, all sort of different intervals. And that, of course, is very distressing for the patient because, they're so, so upset about losing their sense of smell in the first place. And then they think that they've recovered, and then suddenly they're back to square one. I mean, back to square one is a phrase that we hear every day over and over again. So
that is very upsetting, I can imagine.
So what do you make of that?
So we do know that during the process of recovery, the axons have to get back to the brain, to where they belong in the brain, and they tend to go multiple places. They don't always get back to the exact spot where they are supposed to be under normal circumstances. So the messages that the brain is getting are not necessarily going to be interpretable. And there might be certain parts of it missing. So we know, for instance, that there's an olfactory code that requires a certain pattern of receptors to be activated. And that pattern might ideally be spelling brain, B-R-A-I-N. Right? But if, as it's growing back, the B never quite got there, instead of reading brain, you're going to think that it's raining out. Right? So that is how we understand parosmia, most likely. And it is part of this regeneration. So what then happens is there's actually pruning back of those incorrectly targeted axons. So there's this process that can take quite some long period of time. And it's been studied a little bit in animal models, but it's, we can't really study it directly in people. But what we understand is that there's some pruning that can be encouraged and perhaps enhanced through this olfactory training, which is, you've talked about many times. And during that pruning, it might be that there is a period when the intensity drops off prior to the new messages getting more discrete. So again, this is speculation to some degree, to a large degree, because we can't actually watch this happening in real time. Right. From what we know about animal models.
So I would like to add one thing. So in this process of basically rewiring the olfactory epithelium to the brain, so as Nancy said, that could be that the B will also go to the T as well. And so the refining of all this connection is really, at least what we see in mouse, is activity dependent. So more you actually use the olfactory system, more the information will actually, and the neurons in itself, more it will be redefined and basically refined to be able to connect to the right, in the right spot in the brain. So again, that's probably part of the why the olfactory training is so effective.
Exactly. So ladies and gentlemen, just in case you missed it, I think we'll repeat. We've just been listening to why smell training is good from the sort of cell biology perspective. And I think that's such an important message. Nancy, you once, I interviewed you once and you said, smell training is not a far-fetched notion. And it's an interview that I've used over and over again. I think that many people are skeptical about olfactory training, but this idea that olfactory training actually it supports the regrowth of these cells is just very, very important message to get across to patients.
Even more than the regrowth is really the wiring.
The wiring,
yeah.
Correct, getting the correct wiring.
Yep. Okay, so can I I'm going to give you a couple of questions now. So Miriam has asked, so then does this rule out the idea that the smell loss could be our body's defense by shutting down a doorway to the virus? I think this is called the...
Yeah, so that's, I think that the jury is still out somewhat as to whether the virus can get to the brain somehow. But it looks like in some cases where there's neurotropic viruses, where they get into the nerves and the nerves die, that's not what's happening here. Those mature, the cells that detect odors don't have that ACE2 receptor. Now, once the integrity of the cell is damaged by loss of cilia, many things could get into the cell, but they're going to be dying already by that point. So it doesn't seem like that's what's happening, the protection.
Yeah, it's more a collateral damage somehow.
Mm-hmm.
Right.
Okay, and Jane Parker has asked, so in short term, post-COVID anosmia, are the neurons destroyed or is it just the support cells?
It's a combination of the two. So it's triggered by the supporting cells, but then again, because we don't have any scaffold anymore, it's basically the structure is going to be collapsing.
Mm.
And so because it's collapsing and nothing is holding the olfactory neurons up anymore, then also the olfactory neurons are going to probably to die. Now we have some evidences, but of course, this process is always taking long to figure out all the details about it.
Right. Okay, now we have another question from Agata. She says the olfactory epithelium damage is solely caused by virus itself and not at all by our immune reaction to the virus, like macrophages attacking the infected cells in the nose.
Good question, great question, great question. The macrophages are certainly going to be around to clean up the debris from these other cells. And as I mentioned earlier, the supporting cells are important in the defense, and there's some suggestions that they may interact with the immune system in some way. So there may be messages that normally would get through that aren't getting through, or there may be other ways that the immune system is contributing to it, but I think it's going to be a little bit later in the process. Would you agree, Fede?
You know that I like to talk about the chemostasis system.
Yes.
And so basically the chemostasis system is actually driving through a nerve, which is called trigeminal nerve. Whoever knows me is probably already laughing because I managed to say trigeminal nerve at least once. But So the trigeminal nerve is the full nerves that is innervating our face. It's the one that makes you cry when you cut onions, or sneeze when you are, sniffing too hard something with pepper, or all the reaction with wasabi. But this nerve is actually as a protective function as well, and as a pro-inflammatory function. So whenever it's activated, it's actually inducing inflammation. And one of the messengers that actually induce the activation of this system is a small molecule, which is ATP, which is usually used by the cells for energy. But when a cell die, like in our case, the supporting cells are destroyed by the virus, they will release those molecules of energy of ATP in the olfactory epithelium in general. This ATP can then activate the trigeminal system, because this is basically a sentinel for tissue damage. And so our olfactory epithelium needs to be protected by this tissue damage by activating the inflammatory response. Is that clear enough, or I went way
this
too
cycle,
much?
But I think you're going to probably start hearing about this cycle a lot more in the coming months, because it hasn't been really a big part of the conversation up till now, but it's clearly going to be playing an important role in the inflammation, in the inflammatory response and the immune response. And that is how when you have tissue damage, it activates this trigeminal, this sensory irritation pathway, but then that feeds into the immune response and the tissue level immune defense mechanisms.
Yeah. So are you saying that through that mechanism, people are experiencing impairment to their trigeminal nerve? Is that what you're saying? Is that's what's causing trigeminal impairment?
It might be. It might be, everything actually might be correlated. So we have tissue damage starting from the supporting cells. This tissue damage will start activating the system, asking for basically inflammation. But since basically the damage is so big, the inflammation becomes chronic. Mm-hmm. And the chronic inflammation is actually causing even more damage. And that will involve the same system, including the trigeminal system, so chemesthesis. And that's probably why we also see some loss of chemesthesis. And it's also probably, and this is pure speculation, so take it as it comes, but this is probably also why the percentage of loss of chemesthesis is a little bit lower than the loss of olfactory perception, because the damage there has to be really, really strong.
Mm. It's interesting because the issue over cutting onions, that comes up in the group all the time. People are saying the strangest thing, I'm cutting onions and my eyes aren't watering. So this is something that people are definitely noticing.
Yeah.
This is really very unusual. It's very unusual to have a condition that affects this particular pain pathway. And this sensory pathway is very well protected.
But I mean, I'm interested in the sort of chemesthesis, because it seems to be, this is something that I didn't really know very much about before all this happened. And there seem to be a couple of different kinds of trigeminal reactions. So you get, for instance, you can experience wasabi, you can have this sort of tearing of the eyes, the cooling of menthol. Are these things all happening on the same pathway, or is it much more complicated than that? I assume it's more complicated than that.
Yes, that's why I've been working on that for the past 10 years.
Okay.
No, well, and I'm a newbie here. So it's way more complicated. So the reason is the trigeminal nerve, or chemesthesis, is mediated by a wide population of neurons. And those neurons are specialized to detect different kind of stimuli. So we will have a group, which is the one which is mental sensitive, which is also able to detect cold sensation. And this is one group. And then we have the other group, which is the one that reacts, for example, to wasabi or capsaicin. And those are also able to actually drive the inflammatory process. And this is another group. So you have pain perception, but you also have cold perception, and then you have actually hot perception. And this is all depends on basically the kind of receptors they have. So the detecting molecules they express, but also some features of itself, of the neurons in itself. So the ones which are, for example, sensitive to capsaicin and wasabi are the ones able to basically release molecules in the olfactory epithelium, inducing basically inflammation, but maybe also some level of interaction with our olfactory system. Yeah, it's complicated. It's
complicated, but interesting. Okay, I've got some more questions for you now.
Great.
Marie would like to know, do we know why we experience phantosmia? Is there a physiological reason for this, or is it something else? Is this a good sign that neurons are firing or recovering? So phantosmia.
So does she mean phantosmia, do you think, or parosmia?
I don't know. Marie, if you're listening out there, can you quickly go to the Q&A bar and say, yes, I mean phantosmia and not parosmia, because the two are quite different. So parosmia is a qualitative disturbance or a strange alteration in smell from a smell that is present. And with phantosmia, you get a qualitative disturbance where there is no smell present. So maybe Marie will get back to us on that. While we're waiting for you, Marie, we're going to ask a question from Jane, who has said, do you have any idea why the word rain gets through unscathed, but the B of brain doesn't?
Yeah, thanks, Jane. That's another really good question as to which odors may recover or not as we're recovering. And we don't. We do know in mice, there's zones of expression of different types of receptors. And certain types of odors are more strongly detected in certain parts of the epithelium than others. So the damage may be localized. If the damage is localized, you could imagine certain types of odors being more affected than others. In humans, there is a little bit of evidence for some type of organization zonally of the receptors, but we really don't know what the code is. We really don't know the structure in terms of where receptors are for what odors within the nose. So you could imagine that what we see sometimes in other types of damage, closer to the tip of the nose, there's more, if you're breathing in fumes or chemicals or something, it might damage the tissue that's closer to the tip of the nose. Or if it's something that you've swallowed that's going up the back of your nose retronasally, that might damage a certain part of the tissue more than other parts. But right now we can't predict what types of odors are going to come back sooner, or what odors, what types of odor qualities will be most vulnerable to damage. So I think, Jane, some of the work you're doing, it will be interesting to learn as we learn more about what odors people are triggering these conditions, and some of the chemistry, trying to combine some of the chemistry with the biology.
Right. Yeah, I agree with you. I think we've got lots of exciting things to learn on this. Okay, so Marie has written back to us, and she said she was wondering about phantosmia, and she says she struggles with both parosmia and phantosmia, and she's wondering whether phantosmia is the memory of smell somehow being part of the triggering mechanism. I mean, I don't know. I'd love to hear what you say about phantosmia.
So I think of it kind of like people when they talk about phantom limbs, and when people have lost a limb and yet continue to perceive or feel like they're perceiving sensations from that limb. The brain does have a remarkable memory of these perceptions and these sensations. And it's quite feasible, I think, that a message that may be getting through from some other quality, maybe the trigeminal pathway or other chemical field or mechanical sensation as you're breathing air through your nose. We know that mechanical perception of air moving through the nose is an important part of being able to smell. It sort of gates the brain to cue in to odors. So that could be that these sensations that you are still able to perceive, the brain is getting something and saying, oh, I should be smelling something. And so it's grabbing some memory and making that seem real. That's sort of how I think about it.
Right. Good. Thank you. Nancy, while you were talking there, I was just thinking about the quote that you gave to the Wall Street Journal in this article that that came out yesterday and has been widely distributed as, I think, excellent article. And you talked about, so the article covers parosmia and is describing a number of patients in the article. And your comment was, you know, it is perhaps from an evolutionary standpoint, not surprising that these parosmic perceptions are sort of in the danger category. Could you comment on that a bit more, go into a bit more depth on that?
Yeah, so I think a lot about evolutionary terms and what from a survival perspective would make sense. And I think the brain, when it sees information, and this also can happen in hearing things and also with smelling things. If you're smelling something that might be dangerous, your brain will say, oh, that's dangerous. So again, this is sort of how I think about it, that if we're want to survive in an environment and our sense of smell is impaired, but starting to recover, the brain is getting some sort of message, but it's scrambled. The letters are scrambled, like the keyboard that's not right, not put together right yet. And there's some keys missing and we don't necessarily know which ones are missing, but so we're getting letters that are strung together somehow. The brain is going to be grasping to try to make some sense out of that information. And the people that survived are going to be the ones that ran away if it was a dangerous smell, right? And not the ones that thought, oh, this is flowers, when it's actually a toxic chemical. So that's kind of a simplified way of thinking about it, perhaps, but I think the default often in biology, in these processes as we evolved, is the protective reaction. So that's going to be what the brain jumps to first.
And I have to say also from a patient perspective, you know, from the experience of the patient, it's so stressful, this parosmia. And it's like having somebody press the panic button on you, you know, all day, every day. So of course that just makes life unbearable. I think that, you know, this is the kind of the untold story of COVID-19 smell loss, is what these patients who have parosmia are going through. And it is really a very, very challenging situation. I mean, we're collecting stories right now. It's really, some of them are really harrowing. Okay, so we have got a question from Barry Smith. He says, can we speculate on why bathroom smells are found pleasant if it is guarding to notice smells as unpleasant? So as I'm sure many of you are aware, people in the Facebook group say over and over that their perception of fecal odors and also body odor smells is so transformed that those smells start to smell like food and food smells like sewage. And so really would love to know what's going on with that. Got any ideas?
You know, again, what this comes back to is we don't know the code for odor quality. We don't know really, you know, like we understand vision from the wavelength perspective. We can predict if light is this wavelength, it's going to look like that color. We can't predict really from the chemistry to the biology to the perception in olfaction. So it's really still a remarkable mystery. And I think that the experience that we're all learning from, and many of you are going through, that's why the information that you're providing to Chrissy and participating in these studies is so important and so valuable, because we really are, we're learning, we're going to learn, we're going to continue to learn a lot from this experience. If something good can come out of this, it's really going to shed a new understanding on some of these really fundamental questions.
And if I can add something on this, I think it's even more important in the moment we start to actually ask questions during research, because, you know, something like I had no clue about this, I'll be honest. So, you know, knowing that a phenomenon like this is happening, it might really change how I create my hypothesis, how I create my experiment, and how I will gonna address or try to address it in the experimental approach. You know, this is something that, of course, when you are working with animals, where you can do more on the biological, physiological point of view, but you lose a lot on the perception, you cannot do in it. So combining the two things is actually helping in trying to create a more complex and more complete approach to also to the animal study in the end.
Mm-hmm. Good. Okay. I see things in the Facebook group, you know, every day, and I think to myself, I wonder, you know, maybe that's part of the puzzle. Maybe that's part of the puzzle. And certainly, you know, I certainly see trends. I see things that I think, you know, we really have to focus on, like these fluctuations, the fact that we've got late onset parosmia that seems to come out of nowhere, you know, 4 and 5 months down the line. You know, what can that mean when the sense of smell is generally okay? Or people who report perfectly fine sense of smell, and then they get parosmia. So that's not what we would have expected before. So that's there's some really strange things going on.
There is, and I do think it's telling us a lot about how the system is regenerating. Yeah. You know, you could imagine that if I actually had a whole, I was working through and it got too complicated to make the slide, but if you could imagine a sentence that has a number of letters missing, it might still have words. So if we go back to the brain example, if you have the B, but you're missing the I, you're going to get bran, and you're going to think, oh, I'm going to get some fiber, my diet here, it's bran. But maybe it's actually brain, right? So the brain is going to smell like bran. But then maybe you get some other letter coming in there. Maybe an X suddenly regenerated in the middle of the brain. And maybe that X actually belongs over there in the word fox, but at the moment, while it's regenerating, it's stuck in the middle of brain. So you're getting BRXIN or something. Right? So I think it doesn't completely baffle me or surprise me that type of thing happens, because the regeneration process is not going to be necessarily in a perfectly orderly fashion. It's a very complex tissue, and it's a very complex set of wiring diagram that it has to recover to. So it's kind of, you know, not that surprising that there's going to be fluctuations, and at some point, maybe if you were able to test 50 odors every day in some sort of comprehensive test, you learn that, okay, well, it really seemed fine, but actually there's some odors over here that I really can't smell yet, right? And then as they start to regenerate, but maybe in the wrong place initially, that disrupts what you thought you had already. So I think it kind of makes sense to me.
Well, one of the things that comes up a lot, and you've touched on this a little bit before, is we talked about olfactory training and the importance of it. I'm asked a lot if smell training will make the parosmia go away faster.
I think that's a great study to do.
Yeah, and I mean, I know that sort of in an indirect way, smell training will help with the recovery, but will it actually directly affect parosmia? I mean, what do you think?
I mean, I certainly would say that we don't know without the data, but what I would say is it's worth doing the study. Because part of what training does, and this is training for anything, whether you're a flavorist, whether you're training to discriminate wines, or you're training for the ballet or whatever, it's cueing the brain, it's teaching the brain what is important and what's not important, and what it should be focusing on in a given situation. And so any sort of training that you do is going to help the brain to discard what's not relevant and focus on what is relevant. And so I could imagine that it might be helpful, because what you're trying to do is match those perceptions with the memory of what peanut butter smells like, or what have you, right? And so it's going to be trying to make that match, right? And it's going to get closer and closer over time.
So let me ask you about something else that people report a lot, which is that certain not very intense ambient smells can be perceived by people. So they'll say, oh, you know, I could smell this very, very subtle smell. I don't know, let's say that it was, you know, the smell of the soap on my hands the morning after, you know, when I woke up in the morning, I could still smell it, or something that's just really, really subtle, or even the smell of clean laundry, for instance. And yet really smells of high intensity, they're missing. So what is it about some smells that can be perceived at really low concentrations? I mean, you know, even concentrations where they'll say there are people that are living in the same house, they couldn't even smell it, but they could still smell something, you know, floor polish or something that was used, you know, a couple of days before. Why is it that our recovering brains pick out what look like insignificant smells and then grab onto them and make them like really important to us? Fede, you might have an idea.
Yeah, because it's really like neural signal to noise, right, Fede?
Yeah, exactly. So basically, what the brain is usually doing is trying to filter what is signal and what is noise. If there's a lot going on, it might really, the whole brain might actually end up shutting down most of it. The other thing that it's also important, I guess, to consider is there was a study that came out early this year talking about how our brain is actually processing the information coming from the olfactory epithelium, and say that for identification of an odor, you actually don't need the full picture. You just need the essential bits of it. So essential pixel that draw the main figure, and then whatever else comes afterwards is actually it's giving you more idea about the intensity and everything else. So in this situation, I might think that because what it's coming, it's a lot all at the same time, that actually the information becomes too confused to actually being able to be recognized or perceived. And then Another option is, now I bring back in chemesthesis, is actually that odors which are very intense can also activate this trigeminal nerve, so the chemesthesis. So I think many of us might have had, when you smell something like a very strong perfume or something like this, that you like immediately just put your head back, right? And this is because basically your trigeminal nerve is even that is activated by the odors in a way or the other. And from what we said before, that there might be actually, there might be a communication between olfactory system and trigeminal system, because they are both in the olfactory epithelium. And so we don't know exactly what the trigeminal system, so the chemesthesis, does to the olfactory system. I have a couple of ideas, but working on it.
Stay tuned for more research.
Yeah, stay tuned on it. But I think it's really this overload. Yeah, it's really an overload of information most of the time. And also with the parosmia, this idea of how the flow of information gets into our brain, which is important for identification. If you think about that, actually the sequence of the information gets in a little bit scrambled in, because one of the two key informations are not getting in. So the word is completely, from brain, the R is not getting in, or gets in too late, then the information is completely changed.
Right, okay, so just a couple more questions. Believe it or not, the hour's flown by. We had a question about dementia, but I'm going to leave that because this is about the coronavirus today. So we'll be discussing dementia on another occasion, maybe. Why does parosmia distort the smells to a rather strange smell, but why don't we detect bad smells? So Damien, I think we spoke about that a little bit earlier, but we do have a lot of people who report what they call the parosmia smell, the smell. Some people seem to have sort of 3 or 4 parosmia smells, but I think what would you have to say to that, Fede? Would you?
I think it's probably depends really how the wiring has been done. So that probably there are a group of neurons which are all wired or activated similarly, and they basically, the shape that they give or the information that they reveal is more or less always the same.
Yep. We've got another question from Barry, who says, could those with anosmia still have trigeminal activation patterns and think that they can smell the odor? Is the trigeminal activation pattern so sensitive?
There was a now it's old study from Thomas Hummel that he actually showed that some anosmic patients were actually able to distinguish among 2 different odors based on the trigeminal potency. So how the trigeminal nerve was activated. So this depends a lot, as I said before, from concentration. So very strong odors are more likely to activate the trigeminal system. And then the other option is actually really depends from the molecule in itself, how nicely or strongly the trigeminal nerve can detect them. So based on that, you sort of have, if we think about it, you have a different trigeminal sensation from wasabi in comparison to the chili pepper, right? It's different. And so this is actually, or the menthol as well. So those are 3 different sensations. And so those are pieces of information that who unfortunately lack of the olfactory input to help them in will probably grab, and the brain will probably grab to these pieces of information trying to make some sense out of it.
Right. And Fede, is orthonasal trigeminal activation and retronasal trigeminal activation, are they the same, just experienced through a different pathway, or are there differences in the way that works along the tissues, for instance, of the lower nostrils and then the back of the throat?
I think they might be roughly the same, but I'll be fully honest. I don't have full information about it.
It's the same general pathway, but there are differences in which receptors are present. For instance, the
oleocanthal is here down.
The oleocanthal irritation from olive oil is in the back of your throat more.
Right.
Yeah.
Right.
So yeah, I had a very interesting conversation some months ago with actually Paul Breslin, which is working on trigeminal sensation here at Monell. And we talk about how there might be some actually slightly differences in the structure of the same detector, depending on the area where they are. And so that can, again, this is a full hypothesis thrown out there, but this seems so.
Well, I'm sorry to say that I have to stop you now because believe it or not, we've been here for an hour. So thank you very much, Nancy, for joining us, and Fede. It was fantastic having you. In 2 weeks, we'll be having a festive evening. I'll be talking about festive Christmas tipples for those of you who have lost your sense of smell. So something a little bit lighter than what we've been doing for the past couple of months. I hope that you join us then. As always, our webinars are recorded and put up onto YouTube. That might take a couple of days, but you can always catch up, or you can share a link with anyone that is interested. So thank you very much, everyone. Lovely to have you again. Good night.
Good night. Thank you so much.
Good night.